We'll start.
Yeah, ma'am. Please proceed, madam.
Okay. So, hello everyone, and welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class will focus on virology part 2. So, in last class, sir, you asked me one question that whether a virus is living or non-living, and
Yeah,
I said it is living based on its ability to replicate inside a host. So I
yeah,
explained that viruses reproduce by using the host cell's machinery, which shows a key characteristics of living organism. However, I also understand that viruses are considered to be in a gray area because outside the host they do not reproduce or show any metabolic activity and remain inactive, whereas which makes them non-living in that state. So viruses are non-living outside the host body but living inside the host as they replicate. So which is why they are classified as both living and non-living depending on their condition.
yeah, exactly, exactly.
Yeah. Yes, sir. So
Yeah,
I will continue with this virology part 2.
yeah,
Hepatitis Hepatitis viruses are a group of unrelated viruses that primarily infect the liver and cause inflammation. So different hepatitis viruses, that is A, B, C, D, and E, so have distinct structures, transmission route, and clinical outcomes. But all these hepatitis share the ability to cause liver disease, ranging from acute self-limited hepatitis to chronic infection, cirrhosis, or hepatocellular carcinoma. So in this module, we'll focus mainly on hepatitis A and B, which represent RNA and DNA viruses respectively. So these viruses infect the liver and cause distinct clinical pathology by producing characteristic symptoms of jaundice and production of and release of liver enzymes in serum. So most of this disease spread very fast because infected individuals are contagious not only during stage of manifestation of disease but also during the phase of incubation. So hepatitis A virus is a non-enveloped icosahedral RNA virus belonging to the picornavirus family. It contains single-stranded positive sense RNA as its genome. The absence of an envelope makes HIV relatively resistant to environmental stress, including detergents and acids, which help in survive transmission through the fecal oral route. Its morphology, it is a typical enterovirus in the family Picornaviridae. It is small, non-enveloped virus measuring 27 nanometer in diameter. It is also called as enterovirus because HIV leaves to multiply in intestine. And last, it has a single-stranded positive sense RNA genome. Virus structure of HIV. So, hepatitis A virus, this diagram illustrates the structure of hepatitis A by showing its key components and their arrangement within the viral particle. So key parts of the diagram include capsid. The outer layer is the capsid, which is the protein shell protecting the viral genetic material. It is made up of 4 proteins: VP1, VP2, VP3, and VP4 capsid proteins. Single-stranded RNA inside the capsid, it is the single-stranded RNA genome, which is about 7,478 base pair longs. These RNA contain all the genetic instructions for the virus. VPG attached to the RNA is a small protein called VPG, viral protein genome link, which is important for initiating viral DNA replication. Size is particle has a diameter of 27 nanometer. So overall, the diagram presents the hepatitis A virus as a roughly spherical particle consisting of a protein capsid for protection, which encloses an RNA genome for genetic information, and a VPG protein attached to the RNA for replication. So these components together enable the virus to infect host cells and replicate. Viral replication. Once HIV enters the host through ingestion, it travels to the liver via the bloodstream. The virus attaches to hepatocytes and enters through receptor-mediated endocytosis. Being a positive sense RNA virus, its genome directly acts as a messenger RNA for viral protein synthesis. Viral RNA-dependent polymerase then synthesizes complementary RNA strand which serve as a template for new genomes. The newly formed capsids assemble in the cytoplasm and virus particles are released via exocytosis without killing the host cell. Host cell and cultivation. So HAV naturally infect humans and some primates. In laboratory condition, HAV can be cultivated in human cell lines, but replication is often slow and requires specific culture conditions. Since HAV is non-cytopathic, its detection in culture relies on immunological or molecular techniques rather than visible cytopathic effects. It's resistant. HAV is highly resistant compared to many other viruses. It can survive at acidic pH levels, withstand bile salts, and tolerate moderate heat. However, it is inactivated by boiling, chlorination, and formalin treatment. It is sensitive to ether and formaldehyde. Its resistance helps explain its spread in regions with poor sanitation as it can survive for extended periods in contaminated water and food. So, pathogenesis of hepatitis A. First, the virus appears to replicate in GIT and then spreads to the liver. The viruses in liver infect hepatocytes and cause damage to hepatocytes, but the mechanism by which HAV causes cytopathic effect is still not known, whereas cytotoxic T cell appear to cause damage to hepatocytes. Hence, once the infection is cleared, the cell damage is repaired and no chronic infection occurs. Acute hepatitis A. So HAV infection is typically acute and self-limiting. Symptoms begin after an incubation period of 2 to 6 weeks, that is 15 to 45 days, and include fever, fatigue, nausea, vomiting, all this abdominal pain and jaundice. The disease rarely progresses to fulminant hepatitis and chronic infection does not occur. Immunity following infection is lifelong due to the production of neutralizing antibodies. Lab diagnosis. The diagnosis of HIV primarily relies on serological tests. Detection of anti-HIV IgM antibodies indicate recent or ongoing infection, while the presence of IgG antibodies suggests past infection or vaccination. Molecular methods like RT-PCR can also detect viral RNA in stool or serum, which is useful in outbreaks or early infection. So first, detection of virus, one can observe through IEM, immunoelectron microscope. Specimen is feces or urine. Second, isolation part can be isolated on human and monkey cell line. Third is serological. It takes patient blood, separates serum and go for IgM antibodies and anti-HIV. Last is IgG, long period immunoglobulin. Hepatitis B virus. Hepatitis B virus is partially double-stranded DNA virus belonging to the Hepardnaviridae family. It is enveloped spherical and has a unique replication cycle involving reverse transcription. It is an infection disease caused by the hepatitis B virus, which mainly affects the liver. It can cause both acute or chronic infection. Many people have no symptom during the initial stage, whereas the symptom of acute disease are essential for inflammation of the liver with or without jaundice and digestive disorders with nausea and vomiting. Some develop a rapid onset of sickness with vomiting, yellow skin, feeling tired, dark urine, and abdominal pain. These are the underlying issues of hepatitis B virus. Morphology. HBV exhibit remarkable structural complexity. The complete infectious form is known as the Dane particle, which is 42 nanometer in diameter. Surrounding the nucleocapsid is the lipid envelope embedded with surface antigen HBs Antigen inside the nucleocapsid is the viral genome. The genome is small, circular, partially double-stranded, and the viral polymerase enzyme. In addition to DNA particles, infected serum contains a large excess of non-infectious spherical and filamentous particles composed only of HBs surface antigen. These subviral particles play an important role in evading host immune responses by acting as decoys for neutralizing antibodies. Morphology of HBV, it is double-stranded envelope. These are the key components of HBV: HBs AG, HBCAG, DNA polymerase. It contains genome HBEAG, diameter is 42 nanometer. HBV replication. So this diagram depicts the life cycle of a DNA virus inside a host cell, showing the steps from viral entry to the release of new viral particles. So, viral entry. The virus first attaches to a receptor on the host cell surface and enters the cell. Through this interaction, formation of CCC DNA. So what is CCC DNA? It is covalently closed circular DNA. So once inside, the viral DNA is converted into covalently closed circular DNA, that is CCC DNA inside the nucleus. This serves as a template for transcription of a viral mRNAs, which are then exported to the cytoplasm for protein production. Translation: the viral mRNAs are translated by the host ribosome to produce viral proteins required for replication and assembly. DNA synthesis and replication. So, viral DNA polymerase synthesizes the negative DNA strand first, followed by the positive DNA strand synthesis to form new viral genome. And capsidation. So here newly synthesized viral DNA is packed into capsids, a process called encapsidation, budding and secretion. The capsid bud into the membrane of the endoplasmic reticulum, acquiring an envelope as they pass through the cellular membranes. Mature viral particles are then secreted out of the host cell to infect new cells. So, overview, this is a typical pathway for many enveloped DNA viruses where the virus hijacks the host machinery to replicate its genome, produce viral protein, assemble new virons, and exit the cell to perpetuate infection. Incubation time. HBV has a relatively long incubation period, typically ranging from 1 to 6 months. So this long silent phase allows extensive viral replication in the liver before clinical symptoms appear. The incubation period length depends on factors such as the viral load of infection, the individual's immune response, and the route of transmission. For example, perinatal versus blood exposure. Pathogenesis of HBV infection. Hepatitis B virus, after entering the blood, infects the hepatocytes in liver with expression of viral antigen on surface of infected cells. Cytotoxic T cells such as activated CD4 and CD8 lymphocyte. It recognizes various HBV derived protein present on surface of hepatocyte which result in an immunological reaction. Acute hepatitis B virus infection. So in acute infection, individual may present with fatigue, malaise, anorexia, nausea, jaundice, dark urine, and hepatomegaly. The immune response against infected hepatocytes, not the virus itself, is responsible for most of the liver injury. Many acute infection resolve spontaneously as the immune system clears the virus, leading to recovery and lifelong immunity. However, some cases can develop into fulminant hepatitis, which is life-threatening due to massive liver necrosis. This fulminant hepatitis, it occurs in less than 1% of the patients. Chronic hepatitis B virus infection. So, chronic HBV infection occurs when the immune system fails to clear the virus during the acute stage, especially in infections acquired at birth or early childhood. Chronic carriers may remain asymptomatic for years but are at risk of progressive liver damage, cirrhosis, and hepatocellular carcinoma. So, these diseases, long term but rare complications of hepatitis B. Chronic infection is characterized by the persistence of HBsAg for more than 6 months, along with detectable HBV DNA in the blood. Complications of hepatitis B virus infection. Long-term HBV infection is associated with several complications. Cirrhosis results from chronic inflammation and scarring of the liver, impairing its function. Another serious complication is hepatocellular carcinoma, with HBV being a major risk factor for liver cancer worldwide. The integration of HBV DNA into the host genome and ongoing liver cell injury contribute to malignant transformation. Other complications include chronic active hepatitis and hepatic failure. Laboratory diagnosis. Specimen serum is an important specimen because definitive diagnosis of HBV depends on serological testing for HBV infections. Serodiagnosis. Serological markers are crucial for diagnosing and monitoring HBV infection. HBsAg indicates active infection, whether it is acute or chronic. Anti-HBs antibody to surface antigen indicates recovery or successful vaccination. Anti-HBC antibody to core antigen appears in acute infection, persists as IgG in chronic cases. Anti-HBC IgM, so uh, it is marker of active viral replication and high infectivity. Anti-HB E indicates a decline in replication. By interpreting these markers together, clinicians can distinguish between acute, chronic, resolved, and vaccinated stages of infection. Molecular diagnosis. So, molecular methods such as polymerase chain reaction (PCR) are widely used to detect HBV DNA in blood samples. These tests are more sensitive than serological methods and can quantify viral load, which is important for monitoring disease progression and treatment response. Molecular diagnosis is particularly valuable in cases of occult infection and in monitoring antiviral therapy. We'll start with new virus, that is pox viruses. Pox viruses are the largest and most complex viruses known to infect humans. They are unique because they replicate entirely in the cytoplasm rather than in the nucleus. Infections often cause characteristic skin rashes or proliferate lesions. The most well-known member is the variola virus, causes of smallpox, a devastating disease indicated by a global vaccination camp in 1980. Another important member is the Vaccinia virus, used in smallpox vaccines and research. Pox viruses remain medically relevant due to their use in vaccine development and potential zoonotic infections. Properties of pox viruses. Virons, complex structure, oval or brick shape, that is 400 nanometer in length into 230 nanometer in diameter. Its external surface show ridges and it contains core and lateral bodies. Its composition: DNA is 3%, whereas protein is 9% and lipid is 5%. Genome is double stranded DNA. It is linear in shape. Size is 132 375 kilo base pairs, has terminal loops, and has low G plus C content, that is approximately 30 to 40%, except for parapox virus, that is 63%. Proteins include virions that contain more than 100 polypeptides. Many enzymes are present in core, including transcriptional system. Envelope virion assembly involves formation of multiple members. Its replication is cytoplasmic factories. And last is outstanding characteristics, which include largest and most complex viruses, and it is very resistant to inactivation. Virus encoded protein help evade host immune defense system. And last, smallpox was the first viral disease eradicated from the world. Pox virus structure and composition. So, visible as brick-shaped or ellipsoid particles 400 to 230 nanometer. Pox viruses are large, so their genome is double-stranded linear DNA ranging from 130 to 375 kilobase pairs. The virion contains over 100 proteins, including enzyme required for transcription, which is why replication can occur in the cytoplasm without nuclear machinery. The virus is highly resistant to inactivation, and many viral proteins help it evade host immune defenses. Historically, smallpox was the first viral disease eradicated from the world, making pox virus significant in the history of medicine. Pox virus structure, its classification. So it is divided into subfamilies. Pox virus is divided into two subfamilies based on whether they infect vertebrae or insect host. The vertebrate pox viruses fall into 8 genera, with the member of a given genus displaying similar morphology and host range, as well as some antigenic relatedness. Most of the pox viruses that can cause disease in humans are contained in the genera orthopox virus and parapox virus. This is the structure of pox virus. Virus attachment, penetration, and uncoating. Virus particles establish contact with the cell surface and fuse with the cell membrane. Viral cores are released into the cytoplasm. The mRNAs are then transcribed with the viral core and are then released into the cytoplasm. The uncoating protein that acts on the cores is among the more than 50 polypeptides made early after infection. The second stage uncoating step liberates viral DNA from the cores. It requires both RNA and protein synthesis. Replication and maturation of pox viruses. So viral DNA replication occurs in the cytoplasm and appears to be accomplished by viral coded enzymes. Viral DNA replication occurs 2 to 6 hours after infection is discrete areas of the cytoplasm which appear as factories or inclusion bodies in electron micrographs. Maturation, mature virions appear in electron micrographs as a DNA containing core encased in double membranes surrounded by protein and all enclosed within two outer membranes. Some of the particles are released from the cell by budding, but the majority of pox virus particles remain within the host cell. This is the diagrammatic representation of pox viruses. Vaccinia and variola, pox virus infection in humans. So vaccinia virus, it has the agent used for smallpox vaccination is a distinct species of orthopox virus, that is vaccinia virus. Variola has a narrow host range, only humans and monkeys, whereas vaccinia has a broad host range that includes rabbits and mice. Some strains of vaccinia can cause a severe disease in laboratories. Rabbits that has been called rabbit pox. Vaccinia virus has also infected cattle and water buffaloes. And the disease in buffalo is buffalo pox. Both vaccinia and variola viruses grow on the chorioallantoic membrane of the 10 to 12-day-old chick embryo. So, but the later produce much smaller pox. Both grow in several types of chick and primate lines. Pathogenesis and pathology of smallpox virus. So, although smallpox has been eradicated, the pathogenesis of the disease is instructive for other pox virus infection. The portal of entry of variola virus was the mucous membrane of the upper GIT tract, gastrointestinal, upper respiratory tract. After viral entry, the following are believed to have taken place: primary multiplication in the lymphoid tissue, draining the site of entry, transient viremia and infection of reticuloendothelial cells throughout the body, secondary phase of multiplication in those cells leading to a secondary more intense viremia and the clinical diseases. In the pre-eruptive phase, the disease was barely infective by the 6th to 9th days. The disease was barely infective. Lesions in the mouth tend to ulcerate and discharge virus. Later, pustules broke down and discharged virus into the environment of the smallpox patient. So, histopathology Histopathologic examination of the skin showed proliferation of the prickle cell layer. Those proliferated cells contained many cytoplasmic inclusions. There was infiltration with mononuclear cell, particularly around the vessel in the corium. Epithelial cells of the layer become swollen through distension of cytoplasm and underwent ballooning degradation. The vacuoles in the cytoplasm enlarged, the cell membrane broke down and with coalesced with neighboring similarly affected cells, which results in the formation of vesicles. The vesicle enlarge and then become filled with white cells and tissue debris. So thus scarring occurred after variola infection. Similar histopathology is seen with vaccinia, though vaccinia virus ordinarily cause localized pustular lesions only at the site of inoculation. Clinical findings: the incubation period of variola smallpox was 10 to 14 days. The onset was usually sudden. 1 to 5 days of fever and malaise preceded the appearance of the exanthems, which begin as macules, then papules, then vesicles, and finally pustules. These form crust that fell off after about 2 weeks, leaving pink scars that faded slowly. Immunity: an attack of smallpox gave complete protection against reinfection. Vaccination with vaccinia included immunity against variola virus for at least 5 years and sometimes longer. Antibodies alone are not sufficient for recovery for primary pox virus infection. In the human host, neutralizing antibodies develop within a few days after onset of smallpox but do not prevent progression of lesion, and patient may die in the pustular stage with high antibody levels. So cell-mediated immunity is probably more important than circulating antibody. Cell-mediated is very important. Laboratory diagnosis of pox virus. Several tests are available to confirm the diagnosis of smallpox virus, isolation and identification of virus. Here, skin lesions are the specimen of choice for viral isolation. Serology: antibody assays can be used to confirm a diagnosis. Differential diagnosis: because smallpox may be confused with other diseases that give positive result for pox viruses, so differential diagnosis should be made. Treatment: vaccinia immune globin is prepared from blood from persons vaccinated with the vaccinia virus. Methicillin is a chemotherapeutic agent of some value against pox virus. Pseudophore, a nucleotide analog, shows activity against pox viruses in vitro and in vivo. Times of vaccination. Complication of vaccination occurs most commonly under the age of 1 year. Therefore, vaccinating between 1 and 2 years of age is preferable to vaccinating in the first year of life. Revaccination has been done at 3 years of intervals. Third is rabies virus. So these virus transmitted by zoonosis. Rabies virus is the most important member of Rhabdoviridae family, which causes disease in humans. This virus affects the central nervous system, CNS, in cases of inflammation in the brain. Morphology. Bullet-shaped virus size is around 180 to 75 nanometer with one-ended rounded. Its genome is enveloped, linear, and negative single-stranded RNA virus. This is the diagrammatic representation of rabies virus structure. Host range and cultivation. For cultivation, mice can be used, chick embryos can be used, yolk sac, cell lines. The growth can be detected by direct immune fluorescent hemagglutination. Animals are domestic dogs, cats, and wild animals can be used. Resistance. Resistance destroyed in 1 hour at 50 degrees Celsius, destroyed in 5 minutes at 60 degrees Celsius. Sunlight exposure destroyed the rabies virus. UV rays also destroyed. Sensitive to formaldehyde, chloroform, and beta-propiolactone, that is BPL. At room temperature, virus can be destroyed in weeks, whereas at 4 degrees Celsius, it survives for months. Its antigenic properties. G protein, the glycoprotein or G protein, is present on the surface spikes. It is present on the outer lipoprotein envelope of the virion and helps to absorb receptor on the nerve tissue. N protein, nucleoprotein, it is a group of specific antigen. It shows cross reaction with some rabies related viruses. Other antigens include membrane proteins, glycolipid, and RNA dependent RNA polymerase. Antibody produced against this antigen are negative protective. Pathogenesis. Human infection is usually caused by the bite of dogs or other animals. Also, it can cause by licks or aerosols. Entry in the body is once it bites. Rabies is virus deep inside the muscle, saliva, and then multiple in muscle tissue, connective tissue, and reach nerve or neural cells. Finally, it reaches the brain and produce Negri bodies in human. Speed of the virus is 3 minutes per hour, and incubation period is 1 to 3 months depend on the site of bite. This is the diagrammatic representation of pathogenesis for rabies virus infection. Prevention, treatment, and diagnosis. Prevention is through vaccination, both active and passive. Treatment, no specific treatment for this vaccine. Give high sedation in closed, dark, and serene room, no sound. Lab diagnosis, specimen includes saliva, CSF, brain biopsy, urine biopsy of facial skin, and last is cornea. There is two method, direct microscopic, which include immunofluorescence staining technique, AG detection. Second is isolation of virus through cell lines, egg yolk, and mice. Fourth is paramyxovirus. So it includes major respiratory pathogens. These viruses include the most important agents of respiratory infections of infants and young children, as well as the causative agents of two of the most common contagious diseases of childhood, that is mumps and measles. These viruses are the major respiratory pathogens in this age group. All members of this virus family initiate infection via the respiratory tract. Replication of the Respiratory pathogen is limited to the respiratory epithelia, whereas measles and mumps become disseminated throughout the body and produce generalized disease. Properties of this virus, it first include virion. It is spherical in shape, pleomorphic, 150 nanometer in size or more in diameter. It is helical nucleocapsid, that is 13 to 18 nanometer. Its composition is 1% RNA, 73% protein and 20% lipid, and last is 6% carbohydrate. Its genome is single-stranded RNA, linear in shape, non-segmented, negative sense, non-infectious, about approximately 15 kilobase pairs. Protein is 6 to 8 structural proteins is present. Envelope contain viral glycoprotein, that is G protein, H protein or HN protein, which sometimes carries hemagglutinin or neuraminidase activity, and fusion F, that is glycoprotein, and it is very fragile. Replication in cytoplasm, particles bud from plasma membrane. Structure and composition. The morphology of this virus is pleomorphic with particle around 150 to 700 nanometer in diameter, occasionally ranging up to 700 nanometer. The envelope of this virus seems to be fragile, making virus particle labile to storage conditions and prone to distortions in electron micrographs. The viral genome is linear, negative sense, non-segmented RNA about 15 kb in size. Most of this virus contains 6 structural proteins. 3 proteins are complexed with the viral RNA, the nucleoprotein N or NP that forms the helical nucleocapsid, that is 13 to 18 nanometer in diameter and represent the major internal protein, and 2 other large protein designated P and L, which are involved in the viral polymerase activity that functions in transcription and RNA replication. 3 proteins participate in the formation of viral envelope. A matrix M protein underlies this viral envelope. The larger glycoprotein HN or H or G may or may not possesses hemagglutinin and neuraminidase activities and is responsible for attachment to the host cell. It is assembled as a tetramer in the mature virion. The other glycoprotein F fusion mediates membrane fusion and hemolysin activities. The pneumoviruses appears to contain 2 additional small envelope proteins, that is M21 and SH protein. Subfamilies and genera: Paramyxovirinae, Respirovirus, Parainfluenza 1 and 3, Rubella virus, that is mumps P1, 2 and 4, Morbillivirus, that is measles, Henipa virus, that is Hendra and Nipah, Pneumovirinae, that is pneumovirus RSV, metapneumovirus, that is HMPV. Replication cycle: attachment and entry, transcription and replication, and maturation. So attachment, these virus attach to host cell via the hemagglutinin glycoprotein, that is HN, H or G protein. Next, the virion envelope fuses with the cell membrane by the action of fusion glycoprotein F1 cleavage product. Fusion by F1 occurs at the neutral pH of the extracellular environment, allowing the release of the viral nucleocapsid directly into the cell. Transcription, translation, and replication. So, this virus contains a non-segmented negative strand RNA genome. Messenger RNA transcripts are made in the cell cytoplasm by the viral RNA polymerase. Transcriptional regulatory sequences at gene boundaries signal transcriptional start and termination. The most abundant class of transcript produced by an infected cell is from N or NP gene, whereas the least abundant is from the L gene. Viral proteins are synthesized in the cytoplasm. Viral glycoprotein are synthesized in the glycosylated in the secretory pathway. The viral polymerase protein complex PLN is also responsible for viral genome replication. The HN, H, G, and F surface proteins of this virus exhibit minimal antigenic variation over long period of time. It is surprising that they do not undergo antigenic drift as a result of mutation introduced during replication. Maturation. Virus matures by budding from cell surface, progeny, nucleocapsid form in the cytoplasm and migrate to the cell surface. They are attracted to sites on the plasma membrane that are studded with viral HN, H or G and F glycoprotein spikes. The M protein is essential for particle formation, probably serving to link the viral envelope to the nucleocapsid. During budding, most host protein are excluded from the membrane. The neuraminidase activity of the HN protein of parainfluenza viruses and mumps viruses presumably function to prevent self-aggregation of virus particles. Other paramyxoviruses do not possess neuraminidase activities. Mumps. Fifth is mumps virus interaction. Mumps is an acute contagious disease characterized by non-supportive enlargement of one or both salivary glands. Mumps virus mostly cause a mild childhood disease, but in adults, complications including meningitis or orchitis are fairly common. More than one-third of all mumps infection are asymptomatic. Pathogenesis and pathology. Humans are the only natural host for mumps virus. Primary replication occurs in nasal or upper respiratory epithelium. Viremia then disseminates the virus to the salivary glands and other major organ systems. Involvement of the parotoid gland is not obligatory step in infectious process. The incubation period is typically about 14 to 18 days. Virus is shed in the saliva from about 3 days before to 9 days after the onset of salivary gland swelling. Mumps is a systemic viral disease with a propensity to replicate in epithelial cell in virus visceral organs. Virus frequently infects the kidney and can be detected in the urine of most patients. So, the central nervous system is also commonly infected and may be involved in the absence of parotitis. Clinical findings. At least one-third of all mumps infection are subclinical, including the majority of infections in children under 2 years of age. The most characteristic feature of symptomatic cases is swelling of the salivary gland, which occurs in about 50% of patients. Immunity. Immunity is permanent after a single infection. There is only one antigenic type of mumps virus and it does not exhibit significant antigenic variation. Overview of sixth is measles. Measles is an acute, highly infectious disease characterized by fever, respiratory symptoms, and a maculopapular rash. Overview. It is acute, highly infectious disease. Symptoms include fever, respiratory illness, maculopapular rash. Pathogenesis. Humans are the only natural host for measles viruses. The virus gains access to the human body via the respiratory tract, where it multiplies locally. The infection then spreads to the regional lymphoid tissue. Primary viremia disseminates the virus, which then replicates in the reticuloendothelial system. Finally, a secondary viremia seeds the epithelial surface of the body, including the skin, respiratory tract, and conjunctiva. where focal replication occurs. Measles can replicate in certain lymphocytes, which aids in dissemination throughout the body. Multinucleated giant cells with intracellular inclusions are seen in lymphoid tissues throughout the body, that is, lymph nodes, tonsils, appendix, etc. The incubation period is typically lasts 8 to 12 days but may last up to 3 weeks in adults. So during the prodromal phase, that is 2 to 4 days, and the first 2 to 5 days of rash, virus is present in tears, nasal and throat secretions, urine and blood. The rash develops as a result of interaction of immune T cells with virus infected cells in the small blood vessels and lasts about 1 week. Involvement of the CNS is common in measles, that is central nervous system. Clinical findings. The prodromal phase is characterized by fever, sneezing, coughing, running nose, redness of the eye, Koplik spot, and lymphopenia. The conjunctivitis is commonly associated with photophobia, the rash, which starts on the head and then spreads progressively to the chest, the trunk, and down the limbs. Appears as light pink, discrete maculopapules that coalesce to form blotches, becoming brownish in 5 to 10 days. Lymphonia, it is common. Immunity, there is only one antigenic type of measles virus. Infection confers lifelong immunity. Most so-called second attack represent error in diagnosis of either the initial or the second illness. The presence of humoral antibodies indicates immunity. However, cellular immunity appears to be essential for recovery and protection. Laboratory diagnosis. Measles antigens can be detected directly in epithelial cell and respiratory secretions and urine. Antibodies to the nucleoprotein are useful because that is most abundant viral protein in infected cells. Isolation and identification of virus through nasopharyngeal and conjunctiva swabs, blood samples, respiratory secretions, and urine collected from a patient. Serology, that is ELISA, most practical test or method. Treatment and prevention control. Vitamin A treatment in developing countries has decreased mortality and morbidity. Measles virus is susceptible in vitro to inhibition by ribavirin, but clinical benefits have not been proved. A highly effective and safe attenuated live measles virus vaccine has been available since 1963. Seventh is influenza virus. The orthomyxoviridae influenza viruses are a major determinant of morbidity and mortality caused by respiratory disease. An outbreak of infection sometimes occurs in worldwide epidemics. Influenza has been responsible for millions of deaths worldwide. Mutability and high frequency of genetic reassortment and result antigenic changes in the viral surface. Glycoproteins make influenza virus formidable challenges for control efforts. Influenza type A is antigenically highly variable and is responsible for most cases of epidemic influenza. Influenza type B exhibit antigenic changes and sometimes causes epidemics. Influenza type C is antigenically stable and causes only mild illness in immunocompetent individuals. Its property, it is spherical in shape, pleomorphic, that is 80 to 120 nanometer in size. Its genome is single stranded RNA, segmented A and B, 8 segments is present, C is 7 segments. Envelope, it is lipid layer with hemagglutinin, HA, and neuraminidase, Na. Its nuclear transcription and budding maturation. Structure and key points of influenza. There is total 9 structural protein present in influenza virus: NB, PB1, PB2, PA, M1, HA, NA, M2, and last is NS2. Hemagglutinin, it binds virus to host cell and major determinants of the strain variation. Neuraminidase removes salic acid, aids virus release. Genetic resortment, it segment mixing during co-infection and sudden antigenic changes. Replication and antigenic variation through several steps. Attachment and entry, it first binds to salic acid and then endocytotoxic will happen, and then low pH triggers fusion which leads to uncoating. Transcription and translation occurs in nucleus. It uses host mRNA caps. Genome replication is negative sense RNA which leads to positive sense intermediates. Antigenic drift is minor HA-NA changes, seasonal outbreaks. Antigenic shift is major HA-NA changes, that is, new subtypes lead to pandemics. Pathogenesis, immunity, and prevention. Its transmission is through airborne droplets, contact with contaminated surfaces. Pathology, respiratory epithelial destruction, risk of secondary bacterial infection is high. Immunity, HA and NA antibodies are protective. It secretes IgA and serum antibodies, both are important. Treatment include M2 inhibitors and NA inhibitors. Its prevention is inactivated vaccine, updated yearly. Antigenic drift and shift complicates vaccine effectiveness. Lab diagnosis, virus isolation, nasal washing, throat swabs within the days should be done. Serology, detect antibodies that include HA, NA, NP, and M. Last is HIV virus. HIV is nothing but human immunodeficiency virus. The human immunodeficiency virus is a retrovirus that destroys the function of the body's immune system, making the person more susceptible to infections. HIV weakens your immune system by destroying important cells that fight disease and infection. So in early 1981, when discovered in the United States, transmission is through sexual transmission, body fluids, and mother to child. The most frequent mode of transmission of HIV through sexual contact with an infected person. Body fluids HIV transmission is via blood and blood products. HIV, mother to child transmission during pregnancy, during delivery, or breast milk. Its structure, it is spherical in shape. Envelope size is 9 to 120 nanometer. Genome is single-stranded RNA, that is positive sense. Reverse transcription, RNA to single-stranded DNA, then double-stranded DNA, provirus. This diagram illustrates the structure of the human immunodeficiency virus, HIV, which belongs to Baltimore group 6 single-stranded RNA RT viruses. These are single-stranded RNA viruses that use reverse transcriptase to convert their RNA into DNA inside host cell. Glycoprotein, GP120 and GP41. These surface proteins are involved in virus attachment and entry into host cells by binding to receptors on cell surface. Lipid bilayer membrane, a membrane envelope derived from the host cell that surround the virus. Human leukocyte antigen class 1, 1 and 2. Host proteins incorporated into the viral envelope, possibly to help evade the evade immune system. Viral core and proteins. RNA. The virus contains two single-stranded RNA that carry its genetic information. Nucleocapsid protein P24 encases the viral RNA, protecting its genome within the virus. Capsid matrix protein P17 located beneath the envelope. This protein maintains the viral structure. Protease, an enzyme that processes viral proteins to form mature infections virion. Reverse transcriptase, an enzyme that converts viral RNA into DNA once inside the host cell. Integrase, an enzyme that integrates the viral DNA into the host genome for replication, protein P7, P9, other structural or accessory proteins important for viral replication and assembly. In general, this diagram shows the complex architecture of HIV, highlighting its envelope with glycoproteins for host cell targeting, a protective lipid membrane, enzymes essential for viral replication, and proteins that provide structural integrity and functionality to the virus. These components enable HIV to infect human immune cell and replicate within them, causing immunodeficiency over time. Disease progression. The HIV infects CD4 T cells, T helper cell. It enters the CD4 T cells and multiplies. Daughter virus particles are made inside the CD4 T cells and are released into the bloodstream. They infect fresh CD4 T cells. The CD8 T cells kill the virus infected CD4 T cells. Hence, cell population decreases. The main function of CD4 T cell is to fight against the infection of disease producing germs. There are millions of different families of CD4 cells. Each family is designed to fight a specific type of germ when HIV infects these cells. Some of the families of these cells are totally wiped out, hence the ability to fight out certain germs is completely lost. This leads to opportunistic infection. The patient may not suffer from any symptoms in the advanced stages of AIDS during a period ranging between 1 year and 9 years, and perhaps even more so in some cases. But the AIDS virus continue in the meantime, reproduce and multiply itself, as well as the destruction of immune system cells systematically. Symptoms include lymph node swelling, diarrhea, weight loss, fever, cough, shortness of breath. Laboratory diagnosis is PCR. It detects viral RNA or DNA count. Immunology through ELISA, immunolink enzyme sorbent assay, Western blot, and few rapid tests. So summary, we studied about 8 viruses, hepatitis viruses, type A to E, but we only studied in deep 1A and B, cause acute and chronic liver infections diagnosed by serology and molecular tests. Pox viruses, it is the largest DNA virus which include variola, that is smallpox, and vaccinia, replicate in cytoplasm eradicated via vaccination. Paramyxoviruses include measles, mumps, RSV, spread via respiratory route, cause systemic or localized infections. Measles, highly infectious, fever, cough, rash, Koplik spots prevented by live vaccine. Mumps, salivary gland swelling, complications, meningitis, orchitis, lifelong immunity, and post-infection we studied. Influenza, RNA virus with antigenic drift and shift, causes seasonal epidemics and pandemics, vaccine updated yearly. HIV AIDS retrovirus infecting CD4 T cells transmitted sexually via blood, mother to child, leads to immunodeficiency and opportunistic infection. Last is rabies, zoonotic, affects CNS, prevented by vaccination and post exposure. Questions? So for 3 marks, name any 3 viruses included in the Paramyxoviridae family. Second, what is the causative agent of smallpox and how was it eradicated? Third, define antigenic drift in influenza viruses. Fourth, list 2 laboratory diagnostic methods for HIV. Last is mention 2 complications of mumps in adults. For 5 mark questions, first, explain the pathogenesis of measles viruses. Second, describe the replication and antigenic variation of influenza virus. Third, discuss the structure, transmission, and laboratory diagnosis of hepatitis B virus. Fourth, explain the life cycle of pox virus and their clinical significance. And last is describe the progression of HIV infection and its impact on the immune system. So that's all for today. We covered whole topic of viruses.
Yeah, madam. Fantastic. It's excellently covered regarding biology part 2
Yes, sir.
for BSc MLT first year.
Yes, sir. First year it is completed.
First year.
Uh,
Yeah, madam. Thank you so much, madam.
so,
I
yes, sir.
think
Thank
today
you. In
little
next
bit
class,
more, I
we'll
think.
study mycology.
Yeah,
All right, sir.
yeah.
Thank you
Thank
so
you.
much.
Thank you so much.